A new study by University of Maryland entomologists shows that the lifespan for individual honeybees kept in a controlled laboratory environment is 50% shorter than it was in the 1970s. When scientists modeled the effect of today's shorter lifespans, the results corresponded with the increased colony loss and reduced honey production trends seen by US beekeepers in recent decades. Colony turnover is an accepted factor in the beekeeping business as bee colonies naturally age and die off. But over the past decade, US beekeepers have reported high loss rates, which has meant having to replace more colonies to keep operations viable. In an effort to understand why, researchers have focused on environmental stresses, diseases, parasites, pesticide exposure, and nutrition. This is the first study to show an overall decline in honeybee lifespan potentially independent of environmental stresses, hinting that genetics may be influencing the broader trends seen in the beekeeping industry. The study was published in the journal Scientific Reports. The study author Nearman first noticed the decline in lifespan while conducting a study with entomologist associate professor Dennis van Engelsdorp on standardized protocols for rearing adult bees in the laboratory. Replicating earlier studies, the researchers collected bee pupa from honeybee hives when the pupa were within 24 hours of emerging from the wax cells they are reared in. The collected bees finished growing in an incubator and were then kept as adults in special cages. Nearman was evaluating the effect of supplementing the caged bees' sugar water diet with plain water to better mimic natural conditions when he noticed that regardless of diet, the median lifespan of his caged bees was half that of caged bees in similar experiments in the 1970s, 17.7 days today versus 34.3 days in the 1970s. This prompted a deeper review of published laboratory studies over the past 50 years. Although a laboratory environment is very different from a colony, historical records of lab-kept bees suggest a similar lifespan to colony bees, and scientists generally assume that isolated factors that reduce lifespan in one environment will also reduce it in another. Previous studies had also shown that in the real world, shorter honeybee lifespans corresponded to less foraging time and lower honey production. This is the first study to connect those factors to colony turnover rates. When the team modeled the effect of a 50% reduction in lifespan on a beekeeping operation, where lost colonies are replaced annually, the resulting loss rates were around 33%. This is very similar to the average overwinter and annual loss rates of 30% and 40% reported by beekeepers over the past 14 years. The researchers noted that their lab-kept bees could be experiencing some sort of low-level viral contamination or pesticide exposure during their larval stage when they are brooding in the hive and worker bees are feeding them. But the bees have not yet shown overt symptoms of those exposures, and a genetic component to longevity has been shown in other insects such as fruit flies. The next steps for the researchers will be to compare trends in honeybee lifespans across the US and in other countries. If they find differences in longevity, they can isolate and compare potential contributing factors such as genetics, pesticide use, and presence of viruses in the local bee stocks.